Somewhere beneath the waves right now, in a place where no sunlight has ever reached, something is alive that no human being has ever seen. It has no name, it has no classification. It exists in total darkness, under pressure so immense that it would crush a submarine like a paper cup, and it has been living there, thriving there, for longer than anyone can say. No one knows what it looks like, no one knows how it feeds, no one knows how large it is or how small. And the unsettling truth is that we may never know, because the ocean does not give up its secrets willingly. Every single year, marine biologists announce the discovery of new species pulled from the deep sea. Not one or two, not a handful. In some years, the number exceeds 2,000. 2,000 creatures that were previously unknown to science, dragged from the darkness in nets, filmed by remote cameras, or captured in pressurized containers designed to mimic the crushing weight of the abyss. And each time a new species is catalogued, each time a strange translucent body is laid on a laboratory table and examined under light it has never known, the same quiet realization settles over the researchers involved. We have barely begun. The deep ocean, which covers more than 60% of the surface of this planet, remains more than 95% unexplored. That is not a guess. That is not an exaggeration. That is a fact confirmed by every major oceanographic institution on Earth. Think about that for a moment. Humanity has mapped the surface of Mars in greater detail than the floor of its own ocean. We have sent machines to the outer edges of the solar system, yet we have physically visited less than 5% of the seabed beneath us. The water is right there. It surrounds every continent. It fills every basin. And still it remains one of the most alien environments known to exist. Not because we lack the desire to explore it, but because the ocean, at its deepest, is one of the most hostile places on the planet. The pressure alone is enough to kill. The darkness is absolute. The cold is relentless. And life, against every expectation, flourishes. This is the paradox that has haunted marine science for over a century. The deeper you go, the more inhospitable the conditions become. And yet, the deeper you go, the more life you find. Not less, more. Entire ecosystems exist at depths where scientists once believed nothing could survive. Creatures with no eyes, no pigment, no bones, drifting through water columns so vast that the distance from surface to floor exceeds the height of the tallest mountain on land. And every year, those ecosystems reveal something new, something that was always there but simply had not been seen. By the end of tonight, you're going to understand why the deep ocean continues to produce new species at a rate that outpaces every other environment on Earth. You are going to understand why our ignorance of the deep is not shrinking, but in many ways growing. You are going to see how the crushing weight of water, the total absence of light, and the sheer scale of the abyss have conspired to create the single largest reservoir of unknown life on this planet. And you are going to confront a question that is far more unsettling than any creature that might be lurking in the dark. The question is not what is down there. The question is why, after all this time, we still do not know. Before we get started, if you find these mysteries as fascinating as we do, take a second to like the video or subscribe. It's a simple action, but it helps this channel reach more curious minds like yours. The ocean has a way of making the familiar feel alien. Stand on any beach and look out at the water. It seems flat. It seems knowable. But beneath that surface is a world so vast, so cold, so pressurized, and so dark that it makes the surface of the moon look like a neighborhood park. And every year, that world sends us proof that we have not yet earned the right to say we understand our own planet. Now, let's begin. To understand why new species keep appearing from the deep, it is necessary to first understand what the deep actually is. Not as a metaphor, not as an abstraction, but as a physical space with measurable dimensions, real pressures, real temperatures, and real zones of existence that change dramatically as you descend. The ocean is not a single environment. It is a series of stacked worlds, one on top of the other, each with its own rules, its own conditions, and its own inhabitants. And the deeper you go, the stranger those worlds become. The average depth of the ocean is approximately 3,688 meters. That is roughly 12,100 feet. To put that in perspective, the cruising altitude of a commercial aircraft is around 10,000 to 12,000 meters above sea level. The average ocean floor sits at a depth roughly equivalent to the altitude at which most people fly. But you do not feel the weight of the air above you when you are in a plane. In the ocean, you feel everything. Water is roughly 800 times denser than air. Every 10 meters of descent adds approximately 1 atmosphere of pressure to whatever is submerged. At the average ocean depth, the pressure is approximately 368 atmospheres. That means every square centimeter of surface is bearing the force of 368 kilograms. It is the equivalent of stacking several hundred cars on top of a single human body. That is the average depth. The ocean goes much deeper. The deepest known point on Earth is the Challenger Deep, located in the southern end of the Mariana Trench in the western Pacific Ocean. Its depth has been measured at approximately 10,935 meters, or roughly 35,876 feet. At that depth, the pressure exceeds 1,086 atmospheres. If you could somehow lower a steel beam to that depth without protecting it, the pressure alone would compress it. Glass would implode, most metals would buckle, and yet, when the first explorers reached that depth, they found life. Not fossils, not traces, living organisms moving through water that exerts more force per square centimeter than any environment on the surface of the planet. To grasp the sheer vertical scale of the ocean, consider this. If you were to take Mount Everest, the tallest mountain on land, standing at approximately 8,849 meters above sea level, and place it at the bottom of the Mariana Trench, the peak of the mountain would still be submerged under more than 2,000 meters of water. More than a mile of ocean would still sit above the highest point on Earth. That is how deep the ocean goes. That is the scale of the space we are discussing, and almost none of it has been explored by human beings. The ocean is divided into 5 major depth zones, each defined by light, pressure, and temperature. Understanding these zones is essential because each one represents a fundamentally different habitat, and the boundaries between them are where some of the most dramatic biological discoveries occur. The first zone is the epipelagic, also called the sunlight zone. It extends from the surface down to approximately 200 meters. This is the zone most people think of when they imagine the ocean. Sunlight penetrates this layer, photosynthesis occurs, and the vast majority of familiar marine life exists here. Coral reefs, dolphins, tuna, sea turtles, sharks, nearly all of the ocean life that appears in popular culture inhabits this thin skin of water at the very top. But this zone represents less than 5% of the total ocean volume. Everything familiar about the sea lives in a sliver so thin it is almost negligible compared to what lies beneath. Below the epipelagic lies the mesopelagic zone, sometimes called the twilight zone. It begins at 200 meters and descends to roughly 1,000 meters. Sunlight still reaches this zone, but only barely. By 500 meters, the light has faded to a dim blue glow. By 800 meters, it is functionally gone. Photosynthesis cannot occur here. Plants cannot grow. And yet, this zone is one of the most densely populated layers of the ocean. It contains an estimated 10 billion tons of fish biomass, a figure so large that it exceeds the total biomass of all humans on Earth. Most of these creatures are small, many are bioluminescent, and the majority have never been studied in any detail. The mesopelagic is sometimes described as the ocean's hidden engine, because the creatures that live there undertake the largest migration on the planet every single day. Each night, they rise toward the surface to feed. Each morning, they descend back into the dark. This vertical migration involves billions of organisms moving hundreds of meters through the water column. And it has been happening every day for as long as the ocean has existed in its current form. Below the mesopelagic is the bathypelagic zone, beginning at 1,000 meters and extending down to 4,000 meters. This is where the ocean becomes truly alien. No light reaches the bathypelagic. The temperature hovers near freezing, typically between 0 and 4 degrees Celsius. The pressure is immense, ranging from 100 to 400 atmospheres depending on depth. Food is extremely scarce. The only source of nutrition for most organisms in this zone is what scientists call marine snow, a constant slow rain of organic debris falling from the sunlit waters above. Dead plankton, fecal matter, shed scales, decaying tissue. It drifts downward through the water column like a ghostly snowfall, and it is the foundation of nearly all life below the reach of the sun. The bathypelagic zone is vast. It encompasses a water volume greater than all the other zones above it combined. And yet, it has been sampled so infrequently that marine biologists estimate we have observed less than 1% of its total fauna. Expeditions that lower nets or cameras into this zone routinely encounter organisms that cannot be matched to any known species. In some cases, entire families of organisms have been discovered in the bathypelagic that had no previously known representatives. These are not minor variations of known creatures. They are entirely new branches of life, found in water that was assumed, for most of human history, to be lifeless. Beneath the bathypelagic lies the abyssopelagic zone, also known as the abyssal zone. It begins at 4,000 meters and extends down to approximately 6,000 meters. The word abyss comes from a Greek word meaning bottomless, and the name is appropriate. The abyssal zone covers more than 60% of the ocean floor. It is the single largest habitat on Earth by area, and it is almost entirely unexplored. The pressure here ranges from 400 to 600 atmospheres. The temperature is near freezing. The darkness is total and permanent. No human has ever walked on the abyssal plain, and no continuously operated research station exists at these depths. What we know about the abyssal zone comes from brief, expensive expeditions that lower instruments for a few hours at a time, gather what data they can, and then haul everything back to the surface. The creatures of the abyss are adapted to conditions that would be lethal to any surface organism. Their bodies are soft, often gelatinous, lacking the rigid structures that characterize shallow water life. Many have slow metabolisms, growing and reproducing at rates far below those of their surface counterparts. Some abyssal fish can live for more than a century. Others reproduce so infrequently that a single mating event might occur only once in a lifetime. The biology of the abyss operates on timescales that are difficult for surface-dwelling organisms to comprehend. And then there is the Hadal zone, the deepest of all. Named after Hades, the Greek underworld, the Hadal zone begins at 6,000 meters and extends to the absolute bottom of the ocean, the floors of the Great Trenches. There are approximately 46 Hadal environments known on Earth, most of them deep-sea trenches created by tectonic forces. The Mariana Trench, the Tonga Trench, the Kermadec Trench, the Philippine Trench. Each one is a crack in the surface of the planet that descends to depths no other environment can match. The Hadal zone is, in many ways, a series of islands. Each trench is isolated from the others by vast expanses of shallower ocean floor. The organisms that live in one trench may never encounter organisms from another. This isolation has profound biological consequences. It means that life in the Hadal zone evolves independently in each trench, leading to high rates of endemism, the condition in which species are found in one location and nowhere else on Earth. When researchers lower sampling equipment into a Hadal trench for the first time, the probability of encountering species found nowhere else is extraordinarily high. In some expeditions, more than half of the organisms retrieved have been previously undescribed. The scale of these zones, stacked one atop the other, is the first reason that new species continue to emerge from the deep every year. The ocean is not a single room. It is a building with 5 floors, each one larger than the last, each one darker and more pressurized, and each one less explored than the floor above. When scientists say that more than 95% of the ocean is unexplored, they are not speaking in generalities. They are speaking about a measurable volume of water and a measurable area of seabed that has simply never been examined. The history of our attempts to explore these depths is a history of technological limitation. The first serious deep-sea expedition was the HMS Challenger expedition, which set sail from Portsmouth, England, in 1872. Over the course of nearly 3.5 years, the ship travelled approximately 69,000 nautical miles, stopping at 362 observation stations around the globe. Using weighted ropes and dredging equipment, the crew measured ocean depths, collected sediment samples, and hauled up organisms from the deep. The expedition catalogued approximately 4,700 new species. It was a staggering achievement for its time, and it laid the foundation for the science of oceanography. But even the Challenger expedition barely scratched the surface. Its deepest measurement was approximately 8,184 meters, far short of the true ocean bottom. Its collection methods were crude. Nets was lowered blindly. Whatever happened to be caught was brought up. Whatever was too fragile, too small, or too fast to be caught was simply missed. It was not until 1960 that humans first reached the deepest point of the ocean. On January 23rd of that year, Swiss oceanographer Jacques Piccard and United States Navy Lieutenant Don Walsh descended to the floor of the Challenger Deep aboard the bathyscaphe Trieste. The dive took nearly 5 hours. When they reached the bottom, at a depth of approximately 10,916 meters, they peered through a small porthole into water that no human eye had ever seen. Piccard reported seeing a flatfish-like creature on the bottom, though this observation has been debated by marine biologists for decades. What is not debated is the significance of the dive. It proved that life could exist at the absolute deepest point on Earth. It also proved how incredibly difficult it is to get there. After Piccard and Walsh, more than 50 years passed before a human being returned to the bottom of the Challenger Deep. In 2012, filmmaker James Cameron completed a solo dive to the same location aboard the Deep-Sea Challenger submersible. Cameron spent approximately 3 hours on the bottom, collecting samples and filming. He described the environment as a barren, alien landscape, devoid of the dramatic life he had expected. But the samples he brought back contained microbial organisms that were unknown to science. Between these rare manned missions, the deep has been explored primarily by unmanned vehicles. Remotely operated vehicles, known as ROVs, and autonomous underwater vehicles, known as AUVs, have been the primary tools of deep-sea exploration since the 1980s. These machines can descend to depths no human can safely reach and remain there for hours or even days at a time. They are equipped with cameras, sampling arms, sonar systems, and sensors that measure temperature, salinity, and chemical composition. Every major deep-sea discovery of the past 4 decades has been made with the assistance of these vehicles. And yet, even with ROVs and AUVs, the coverage is minimal. The total area of ocean floor that has been visually surveyed by cameras or submersibles is estimated to be less than 0.05%. That is not 5%. That is 0.05%. To put this another way, for every square kilometre of ocean floor that has been photographed, there are roughly 2,000 square kilometres that have never been seen by any camera, human or robotic. The mathematics of deep-sea exploration are sobering. The ocean floor covers approximately 361 million square kilometres. A typical ROV dive covers a strip of seafloor a few meters wide and a few kilometres long. Even if you were to run 1,000 ROV dives a year, each covering 10 square kilometres of seafloor, it would take more than 36,000 years to survey the entire ocean bottom. And that calculation assumes you only need to look once at one depth in one season. The ocean is not static. Its currents shift, its temperatures fluctuate, its inhabitants migrate, breed, and die. A single survey tells you what was in one place at one time. It tells you almost nothing about what might be there tomorrow. This is why new species continues to be found, not because they are new to the ocean. They have been there all along. They are new to us. We are the ones arriving late, peering into a world we have barely begun to map, pulling up handfuls of life from a body of water that contains more volume than all the land habitats on Earth combined. Every expedition is a first glimpse. Every sample is a single grain of sand lifted from a beach that stretches farther than the eye can see. But the sheer size of the ocean is only part of the explanation. Something else is at work in the deep. Something that makes it not just large, but fundamentally resistant to the kind of exploration that has mapped the rest of the planet. And this is where the answer becomes deeply unsettling. The central problem with deep-sea exploration is not simply that the ocean is large. It is that the ocean actively resists being explored. Every physical property of the deep works against human technology, human biology, and human patience. The pressure destroys equipment. The darkness blinds cameras. The cold degrades batteries. The distance from the surface makes communication slow and unreliable. And the organisms that live in these conditions are so exquisitely adapted to their environment that the act of bringing them to the surface often kills them. We are not merely looking at a large space that needs more time to map. We are looking at an environment that fights back. Consider the problem of pressure. At sea level, the atmosphere exerts a pressure of 1 atmosphere on every surface. This is the pressure that human bodies are designed to withstand. For every 10 meters of descent into the ocean, the pressure increases by 1 additional atmosphere. At 100 meters, the pressure is 11 atmospheres. At 1,000 meters, it is approximately 101 atmospheres. At 4,000 meters, the average depth of the abyssal plain, the pressure is roughly 401 atmospheres. At the bottom of the Mariana Trench, as mentioned earlier, it exceeds 1,086 atmospheres. This means that any instrument sent to the deep must be built to withstand forces that would destroy most materials on the surface. The housings for deep-sea cameras are typically made of titanium alloy, machined to tolerances measured in fractions of a millimeter. A single flaw in a pressure housing, a hairline crack, a poorly seated seal, will result in catastrophic implosion. The housing does not leak. It collapses instantaneously, crushed by the weight of the water above it. The cost of building equipment that can survive these pressures is enormous, and the cost of losing that equipment to a single failure is equally enormous. This is one reason why deep-sea exploration remains the province of a small number of heavily funded institutions. A single deep-rated ROV can cost between $1 and $10 million. The ship required to deploy it costs tens of thousands of dollars per day to operate. A single expedition to a deep-sea trench can cost more than $1 million, and it may return with only a few hours of footage and a handful of specimens. Compare this to terrestrial biology, where a researcher with a pair of binoculars and a notebook can survey a forest for weeks at minimal cost. The economics of deep-sea exploration are punishing, and they ensure that the vast majority of the ocean will remain unvisited for the foreseeable future. But the problem of pressure extends beyond equipment. It also affects the organisms themselves. Many deep-sea creatures are adapted to high pressure at the molecular level. Their cell membranes contain different lipid compositions than surface organisms, allowing them to remain fluid and functional under forces that would solidify normal cell membranes. Their proteins are shaped differently, folded in ways that maintain function under compression. When these organisms are brought to the surface, the pressure drop causes their cells to expand, their membranes to destabilize, and their proteins to unfold. Many deep-sea specimens arrive at the surface dead or dying, their bodies distorted by decompression. This makes it extraordinarily difficult to study them alive. It also means that the specimens available to taxonomists are often damaged, incomplete, or altered in ways that make classification unreliable. In recent years, pressurized aquaria have been developed that can maintain deep-sea organisms at something approximating their native pressure. These systems are complex, expensive, and small. They can house a few organisms at a time, not entire ecosystems. And maintaining them requires constant monitoring, because even minor fluctuations in pressure can be lethal to the inhabitants. The technology is improving, but it remains in its infancy. For most deep-sea organisms, the act of being discovered is also the act of being destroyed. Now consider the problem of darkness. Below approximately 1,000 meters, the ocean is entirely without natural light. There is no faint glow, no residual luminescence from the surface. The darkness is absolute and permanent. For organisms that live in this environment, light is either meaningless or actively threatening. Many deep-sea creatures are bioluminescent, producing their own light through chemical reactions in specialized organs called photophores. This light serves various purposes, from attracting prey to communicating with potential mates. But the light produced by deep-sea organisms is faint, carefully controlled, and specific to narrow wavelengths. The powerful floodlights mounted on submersibles and ROVs are, from the perspective of these creatures, an alien intrusion of staggering intensity. Organisms that have evolved in total darkness are suddenly exposed to thousands of lumens of artificial light. Many flee, many freeze, many are blinded. The very tools we use to see the deep actively disrupt the behavior of the animals we are trying to observe. This creates a fundamental observational paradox. We cannot see in the deep without bringing light, but bringing light changes the behavior of everything in the deep. Researchers have long suspected that the organisms most frequently captured by submersible cameras are not representative of the deep-sea community as a whole. They are the ones that happen to be near the vehicle, that did not flee from its lights, or that were attracted to its illumination. The shy ones, the fast ones, the ones that respond to light by swimming away as quickly as possible. These organisms may be grossly underrepresented in our records. We may be cataloguing only the most tolerant fraction of deep-sea life and missing everything else. The problem of temperature compounds these difficulties. The deep ocean is cold. Below the thermocline, a boundary layer that separates warm surface water from cold deep water, temperatures drop to between 0 and 4 degrees Celsius and remain there permanently. Organisms adapted to these temperatures have metabolisms that are extremely slow by surface standards. They grow slowly, reproduce slowly, and move slowly. Some deep-sea fish take decades to reach reproductive maturity. Some deep-sea corals grow less than a millimeter per year. A colony of black coral pulled from deep water near Hawaii was estimated to be more than 4,000 years old based on radiocarbon analysis. 4,000 years. That coral was alive when the Egyptian pyramids were being built, and it was still growing on the ocean floor when it was collected. The slowness of deep-sea life has a direct bearing on why species continue to be discovered. Slow reproduction means slow evolution, which means that species can persist for extremely long periods without significant change. But slow reproduction also means that populations remain small and scattered. A species with a reproductive cycle measured in decades will not produce the enormous populations that characterize fast-reproducing surface organisms. Instead, deep-sea species often exist as small, widely dispersed populations spread across vast areas of seafloor. Finding them requires either extraordinary luck or extraordinary effort. A net dragged across a patch of abyssal plain might capture one individual of a species that has a population of only a few thousand spread across millions of square kilometers. The next expedition to the same area might find nothing. The one after that might find a different species entirely. Each sample is a snapshot of an impossibly large and sparsely populated world. And it gets worse. The deep ocean is not a uniform environment. It is riddled with features that create isolated habitats, each capable of supporting unique communities of organisms. Seamounts, underwater mountains that rise from the ocean floor but do not reach the surface, number in the tens of thousands across the global ocean. Some estimates suggest there are more than 100,000 seamounts taller than 1,000 meters, and the total number, including small surf features, may exceed 1 million. Each seamount is a potential island of life, surrounded by the relative emptiness of the abyssal plain. Currents deflect around seamounts, creating upwellings that bring nutrients to their peaks. Organisms colonize these peaks, forming communities that may be isolated from other seamount communities by hundreds or thousands of kilometers. This isolation drives speciation, a population of organisms on one seamount, separated from its nearest relatives by a vast stretch of empty deep ocean, will, over time, diverge into a distinct species. The same principle applies to hydrothermal vents, cold seeps, whale falls, and the other patchy habitats that dot the deep-sea floor. Each one is an oasis in a desert of mud and cold water. Each one supports a community of organisms found nowhere else, and each one has the potential to harbor species that have never been seen by human eyes. Hydrothermal vents deserve particular attention because their discovery in 1977 fundamentally changed our understanding of what life requires. Before the discovery of hydrothermal vents, all known ecosystems on Earth were ultimately dependent on sunlight. Photosynthesis powered the base of every food web. Even deep-sea ecosystems were assumed to depend on marine snow, that rain of organic matter from the sunlit surface. The discovery of vents on the Galapagos Rift by the research submersible Alvin and a team of geologists from Oregon State University and the Woods Hole Oceanographic Institution revealed something entirely unexpected. Here were thriving ecosystems dense with life, clustered around cracks in the seafloor where superheated, mineral-laden water gushed from the Earth's interior. The water emerging from these vents reached temperatures of more than 350 degrees Celsius. It was laden with hydrogen sulfide, methane, and dissolved metals. And surrounding these vents, in water that was toxic by every surface standard, were fields of giant tube worms, clusters of mussels, swarms of shrimp, and mats of bacteria so thick they looked like carpets. The key to these ecosystems was chemosynthesis, a metabolic process in which bacteria use chemical energy from hydrogen sulfide and other vent chemicals to produce organic matter. These bacteria form the base of the food web at hydrothermal vents, replacing the role that photosynthetic organisms play at the surface. The entire ecosystem is powered not by the sun, but by the heat and chemistry of the Earth itself. This was a revelation. It meant that life could exist without sunlight. It meant that ecosystems could be self-sustaining in total darkness. And it meant that the deep ocean contained a source of energy for life that was entirely independent of anything happening at the surface. Since their initial discovery, hydrothermal vents have been found on mid-ocean ridges around the world. Each vent field supports a unique community. The organisms at vents in the Atlantic are different from those in the Pacific. The organisms at shallow vents differ from those at deep vents. And because vents are ephemeral, lasting from a few decades to a few centuries before the underlying geological activity shifts and the vent goes cold, the organisms that depend on them must be capable of dispersing to new vents or face extinction. This creates a constant cycle of colonization, adaptation, and speciation that generates biological novelty at a rate far higher than the relatively stable environments of the surface ocean. The same is true of cold seeps, locations on the ocean floor where methane, hydrogen sulfide, and other hydrocarbon-rich fluids seep slowly from beneath the sediment. Cold seeps are found on continental margins around the world, often at depths of several hundred to several thousand meters. Like hydrothermal vents, they support chemosynthetic communities that are independent of sunlight. But unlike vents, cold seeps can persist for thousands of years, creating stable habitats that allow communities to develop over much longer timescales. The tube worms found at some Gulf of Mexico cold seeps have been estimated to live for more than 250 years, making them among the longest-lived animals on Earth. Then there are whale falls, the carcasses of dead whales that sink to the ocean floor. A single whale carcass can weigh dozens of tons and provides a concentrated source of nutrition in an environment where food is otherwise scarce. When a whale falls to the deep sea floor, it initiates a succession of ecological stages that can last for decades. First, mobile scavengers arrive, hagfish, sleeper sharks, and amphipods, consuming the soft tissue over a period of months to years. Then, enrichment opportunists colonize the remaining bones and sediment, feeding on the organic matter that has leached into the surrounding area. Finally, the bones themselves become hosts for sulfophilic communities, bacteria and animals that feed on the hydrogen sulfide produced by the decomposition of bone lipids. This final stage can last for 50 to 100 years. Each whale fall is a temporary island of abundance, and each one supports organisms that may not be found anywhere else. Several species of bone-eating worm in the genus Ostax have been discovered exclusively on whale falls. These worms have no mouth, no gut, and no anus. They feed by dissolving bone using acid secreted from specialized root-like tissues, relying on symbiotic bacteria to extract nutrients from the dissolved lipids. Before the first whale fall was discovered in 1987 by researchers from the Scripps Institution of Oceanography, no one knew these worms existed. Since then, more than 25 species of Ostax have been described, and new ones continue to be found with nearly every new whale fall studied. The picture that emerges is one of staggering complexity. The deep ocean is not a single barren plain. It is a mosaic of habitats, each supporting its own community of organisms, many of them found nowhere else. Seamounts, vents, seeps, trenches, whale falls, abyssal plains, canyon walls, each one is a potential reservoir of undiscovered life. And because these habitats are separated by enormous distances, the organisms in each one tend to evolve independently, producing unique species that exist in only one location. But here's what makes this truly terrifying. The rate at which we are discovering new deep-sea species is not increasing because we are getting better at looking. It is increasing because, with each new technology and each new expedition, we are looking in places that have never been looked at before, and every time we look somewhere new, we find something new. The implication is profound. We are not running out of new species to find. We are not approaching the bottom of the list. Every new look reveals that the list is longer than we thought. Over the past half-century, a series of expeditions, technologies, and scientific campaigns have tried to make sense of the deep ocean's biological diversity. Each one has advanced our understanding, and each one has, in its own way, demonstrated just how far we remain from a complete picture. The deep resists cataloguing, it resists understanding, and it has a consistent habit of overturning assumptions that were considered settled. Consider the case of hydrothermal vent fauna. After the initial discovery of vent ecosystems in 1977, researchers expected that vent communities around the world would share similar species. The reasoning was straightforward. Hydrothermal vents are found along the global mid-ocean ridge system, a continuous chain of underwater mountain ranges that encircles the planet like the seam of a baseball. Since vents occur on this connected system, it seemed reasonable that organisms could migrate along the ridge from one vent field to another, maintaining genetic connectivity across ocean basins. The early discoveries seem to support this. Giant tube worms of the species Riftia pachyptilla were found at multiple vent sites in the eastern Pacific. The same species of vent shrimp appeared at sites separated by hundreds of kilometers on the mid-Atlantic ridge. But as more vent fields were explored, the picture became far more complicated. Vents in the Indian Ocean, first explored in 2001 by a team aboard the German research vessel Sun, were found to support communities that bore almost no resemblance to those in the Pacific or Atlantic. New species of snails, new species of crabs, new species of polychaete worms, none of them previously known. Vents in the Southern Ocean near Antarctica, discovered in 2012, were equally alien. The dominant organisms were species of yeti crab, furry white crustaceans, with dense mats of filamentous bacteria growing on their claws. These crabs were unknown before 2005, when the first species, Kiwa hirsuta, was described from a vent site southeast of Easter Island. Since then, multiple species of yeti crab have been identified, each from a different vent field, each distinct. The pattern is consistent. Every new vent field explored yields a significant proportion of new species. A review published in the journal Progress in Oceanography in 2016 compiled data from more than 300 vent sites around the world and concluded that approximately 646 species of animals were known from hydrothermal vents at that time. Of these, more than 70% were found at only one or two vent sites. The level of endemism was extraordinary. It meant that the vast majority of vent species had extremely limited distributions, existing in tiny patches of habitat surrounded by thousands of kilometers of lifeless deep ocean. But here's the problem. The global mid-ocean ridge system extends for approximately 65,000 kilometers. The total number of known hydrothermal vent fields, as of the most recent surveys, is fewer than 700. The actual number of active vent fields is estimated to be far larger, potentially several thousand, with some models suggesting as many as 5,000 or more. If each vent field supports a community with high endemism, and most vent fields have never been explored, then the total number of undiscovered vent species is likely to be in the thousands, possibly the tens of thousands. The numbers are striking. Each vent is a potential source of species found nowhere else, and most vents have never been visited. The attempt to explore brine pools presents a different set of challenges. Brine pools are one of the most extraordinary features of the deep ocean, found on the floor of the Gulf of Mexico, the Red Sea, the Eastern Mediterranean, and other locations where ancient salt deposits underlie the seafloor. Brine pools are lakes of hypersaline water that sit on the ocean floor. The brine is so much denser than the surrounding seawater that it forms a distinct layer, complete with a visible shoreline and a surface that ripples in the current. Submersibles can descend to the edge of a brine pool and observe the boundary between normal seawater and brine as clearly as you would observe the boundary between water and air at the surface. The conditions within brine pools are extreme even by deep-sea standards. The salinity can be 3 to 8 times that of normal seawater. The concentrations of methane and hydrogen sulfide are often high enough to be toxic to most organisms. Yet the shores and surfaces of brine pools support dense communities of chemosynthetic mussels, bacterial mats, and polychaete worms. Some organisms appear to live partially submerged in the brine itself, tolerating conditions that would kill almost any other animal on Earth. The biology of brine pools remains poorly understood. Most known brine pools have been explored only briefly, during single or handful of submersible dives. The deepest and most remote brine pools, particularly those in the Red Sea, at depths exceeding 2,000 meters, have barely been studied at all. In 2020, a research expedition to the Red Sea discovered a new brine pool at a depth of approximately 1,770 meters. Initial sampling revealed bacterial communities that were genetically distinct from those found at any other brine pool. The chemistry of the pool was also unique, with concentrations of dissolved metals that had not been seen at other sites. Every new brine pool found represents a new biochemical environment, and every new biochemical environment has the potential to support new forms of life. The mystery of the deep sea's biological diversity extends beyond individual habitats. There are entire categories of organisms that have been discovered in the deep, only to raise more questions than they answer. Consider the case of xenophytophores, giant single-celled organisms found on the abyssal and hadal ocean floors. Some xenophytophores reach sizes of more than 20 centimeters across, making them among the largest individual cells on Earth. They build their their external structures, from bits of sediment, sponge spicules, and the skeletal remains of other organisms, cementing them together with organic glue. The result is a structure that looks like a crumpled piece of wet paper sitting on the ocean floor at depths of 4,000 to 10,000 meters. Xenophytophores are found worldwide, but their taxonomy is in a state of considerable confusion. The total number of described species is only about 75, but researchers who specialize in these organisms suspect that this number represents only a fraction of the true diversity. The problem is that xenophytophores are extremely difficult to collect intact. They are fragile, falling apart at the slightest touch. They disintegrate during conventional trawling, and because they live at extreme depths, they can only be collected by specialized, gentle sampling equipment, which is rare and expensive. A deep-sea camera lowered to the abyssal plain may photograph dozens of xenophytophores in a single dive, many of them clearly different from any known species, but photographing them and collecting them are very different things. Without physical specimens, formal species descriptions are impossible under current taxonomic rules. The deep sea is full of organisms that have been seen but cannot be named. A similar story unfolds with the creatures of the Hadal trenches. The Hadal zone, as discussed earlier, comprises approximately 46 known deep-sea trenches. Of these, fewer than half have been biologically sampled in any meaningful way. The most extensively studied are the Mariana Trench, the Kermadec Trench, the Tonga Trench, the Japan Trench, and the Pruchile Trench. Even in these relatively well-studied systems, new species are found on virtually every expedition. The dominant animals of the Hadal zone are amphipods, small crustaceans that swarm in enormous numbers at the bottoms of the deepest trenches. When researchers lower baited camera systems to Hadal depths, the images that return are striking. Within minutes of the bait arriving on the bottom, hundreds or thousands of amphipods appear, converging from every direction, drawn by the scent of food in an environment where food is vanishingly rare. Some of these amphipods are enormous by the standards of their kind, reaching lengths of more than 30 centimeters. This phenomenon, known as gigantism, is not well understood. It may be related to the extreme pressure, the cold temperatures, or the scarcity of food, which favors organisms that can store large amounts of energy in their bodies. What is known is that Hadal amphipods are extraordinarily diverse. A study published in the journal Deep Sea Research in 2019 analyzed amphipod populations from 5 Pacific trenches and found that each trench contained its own suite of species. The overlap between trenches was minimal. Species found in the Mariana Trench were absent from the Kermadec Trench, and vice versa. The trenches, despite sharing similar depth ranges and physical conditions, had evolved largely independent communities of amphipods. The isolation of the Hadal zone, the fact that each trench is a self-contained environment cut off from its neighbours by shallower ocean floor, appears to drive rapid speciation. This finding has been replicated for other Hadal organisms. Snailfish, the deepest living vertebrates known, have been found at depths exceeding 8,000 meters in multiple trenches. Each trench appears to harbor its own species of snailfish, adapted to the specific conditions of that particular trench. The Mariana snailfish, Pseudolaparis swirae, described in 2018, is found only in the Mariana Trench. It was discovered at a depth of approximately 7,966 meters, making it the deepest fish ever documented. Its body is pale and translucent, its bones partially dissolved into cartilage, and its muscles infused with high concentrations of a chemical compound called trimethylamine N-oxide, which stabilizes proteins under extreme pressure. Nothing like it exists anywhere else on Earth. The Yonaguni Monument, a massive rock formation discovered off the coast of Yonaguni Island in Japan in 1986, introduces yet another angle of deep-sea mystery. Found at a depth of approximately 25 meters, it is not a deep-sea feature by the standards we have been discussing, but its discovery sparked a debate that illustrates a broader truth about underwater exploration. Marine geologist Masaki Kimura studied the formation extensively and argued that its regular geometric shapes, including flat terraces, sharp edges, and right-angle steps, were evidence of human construction, possibly a structure built by an ancient civilization and subsequently submerged by rising sea levels. Other geologists countered that the formation could be explained by natural geological processes, specifically the fracturing of sandstone along natural bedding planes. The debate remains unresolved. But what is significant, for our purposes, is that the Yonaguni Monument sat on the ocean floor for an unknown length of time before being noticed by a sport diver. It was not found by a scientific survey. It was not detected by sonar. It was stumbled upon by accident. And it lies in shallow, accessible water, just 25 meters down, in a region that has been inhabited by humans for thousands of years. If a formation that large, that distinctive, and that shallow can go unnoticed for so long, what is hiding in the abyssal plain at 4,000 meters? What is living in the Hadal trenches at 10,000 meters? The deep ocean does not reward the casual observer. It punishes carelessness, it ignores ambition, and it keeps its contents hidden behind barriers of pressure, darkness, and distance that no amount of human determination has yet overcome. The attempts to solve this problem through technology have been creative, expensive, and only partially successful. Environmental DNA sampling, known as eDNA, is one of the most promising recent innovations. The concept is simple. Every organism sheds DNA into its environment through skin cells, mucus, waste products, and decomposition. This DNA persists in the water for hours to days before degrading. By collecting water samples and analyzing the DNA they contain, researchers can detect the presence of organisms without ever seeing or catching them. A single water sample from the deep sea can contain DNA from dozens or hundreds of different species, many of them previously unknown. The power of eDNA was demonstrated dramatically in a study published in 2022, which analyzed water samples collected from the Mariana Trench. The study detected DNA from more than 180 species, including fish, crustaceans, and soft-bodied organisms, many of which could not be matched to any known species in genetic databases. The implication was clear. The Mariana Trench, despite being the most famous and arguably the most studied deep-sea feature on Earth, still contains a substantial community of organisms that have never been identified. But eDNA has limitations. It can tell you that an organism's DNA is present in a water sample. It cannot tell you what the organism looks like, how it behaves, how it reproduces, or how it fits into the ecosystem. A strand of DNA floating in cold dark water does not carry with it a picture of the creature it came from. For formal species descriptions, for understanding ecology, for conservation, physical specimens are still required, and obtaining physical specimens from the deep remains as difficult and expensive as ever. Autonomous underwater vehicles represent another approach. Modern AUVs can descend to depths of 6,000 meters or more, navigate pre-programmed routes along the ocean floor, and collect data for days at a time without human intervention. Some are equipped with cameras, others with sonar systems that can map the seafloor with centimeter-scale precision, and still others with water sampling systems for eDNA collection. But AUVs are limited by battery life, by the vile speed at which they can cover ground, and by the fact that they cannot collect physical specimens. They are eyes without hands. They can show us what is there, but they cannot bring it back. The deep-sea mining debate has added a new dimension to these challenges. Several companies and national governments have proposed mining the deep-sea floor for polymetallic nodules, metal-rich deposits the size of potatoes that litter vast areas of the abyssal plain. These nodules contain manganese, nickel, cobalt, and copper, metals that are critical for modern electronics and renewable energy technologies. The most extensive deposits are found in the Clarion-Clipperton Zone, a region of the eastern Pacific Ocean spanning approximately 4.5 million square kilometers between Hawaii and Mexico. In advance of potential mining operations, environmental impact assessments have been conducted across the Clarion-Clipperton Zone. These assessments have revealed astonishing levels of biological diversity. A study published in 2023 reported that of more than 5,000 species identified in the Clarion-Clipperton Zone, approximately 88 to 92% were new to science. That figure bears repeating. Nearly 90% of the species found in one of the most studied deep-sea mining regions on Earth had never been described before. The organisms lived in and on the very nodules that mining companies plan to collect. Removing the nodules would remove their habitat. Describing the organisms before the habitat is destroyed became a matter of urgent scientific concern. The math is striking, even in an area that has been the subject of intensive scientific study for more than a decade. Motivated by an industry willing to spend billions of dollars, the majority of species remain undescribed. The pace of taxonomy, the slow, careful work of describing and naming species, cannot keep up with the pace of discovery. There are not enough taxonomists, not enough funding, and not enough time. The deep ocean is producing new species faster than science can process them. Something is deeply wrong with our assumptions about how well we know our own planet. We speak of the Earth as explored. We see satellite images of its surface and believe we have seen everything. But the satellite sees only the surface of the water. It cannot see the abyssal plain. It cannot see the Haddle Trench. It cannot see the hydrothermal vent, or the cold seep, or the whale fall. The explored Earth is the dry Earth, the thin crust of land that represents less than 30% of the planet's surface. The rest is water, and beneath that water lies a world we have barely entered. The deeper truth about the discovery of new species in the deep ocean is not a story about fish or worms or microbes. It is a story about the limits of human knowledge and the size of human ignorance. We are a species that has spent centuries cataloguing the life on land. We have named approximately 2 million species of plants, animals, fungi, and microorganisms. We have filled museums with specimens, libraries with descriptions, and databases with genetic sequences. And yet, by the most conservative estimates, the total number of species on Earth may exceed 8 million, with some estimates reaching 12 million or more. The gap between what we have named and what exists is enormous, and the deep ocean is where the largest portion of that gap is concentrated. Let that sink in. More species may be unnamed on Earth right now than have been named in the entire history of science, and the majority of those unnamed species are likely to be found in the deep sea. The Census of Marine Life, one of the most ambitious biological surveys ever undertaken, ran from 2000 to 2010. It involved more than 2,700 scientists from 80 countries, conducting 540 expeditions across every ocean basin. Over the course of a decade, The census identified more than 6,000 new species, bringing the total number of known marine species to approximately 250,000. But the census also estimated that the total number of marine species on Earth could be as high as 1 million or more, 250,000 known out of a possible 1 million. The deep sea, which constitutes the largest marine habitat by volume, was identified as the primary reservoir of undiscovered diversity. The reasons for this should by now be clear. The deep ocean is immense, dark, cold, pressurized, and sparsely explored. But there is an additional factor that is less often discussed but equally important. The deep ocean is old. Not old in the sense that a forest is old or a mountain range is old. Old in a way that is almost incomprehensible. The deep ocean, as a continuous body of cold, pressurized water, has existed in something resembling its current form for many millions of years, according to geological models. During that time, life in the deep has had an unimaginably long period in which to diversify, adapt, and specialize. The surface of the Earth has undergone dramatic changes during this time: ice ages, volcanic eruptions, mass extinctions, continental rearrangements. Many of these events devastated surface ecosystems, but the deep ocean, buffered by its immense volume and its distance from the surface, was affected far less severely. This stability has profound implications. On land, mass extinction events periodically reset the biological clock, wiping out large proportions of species and opening ecological niches for new ones to fill. In the deep sea, the biological clock has been running more continuously. Species that might have gone extinct on land persisted in the deep. Lineages that diversified on the surface and then disappeared may still have deep-sea representatives. The deep ocean is, in a very real sense, a biological archive, a reservoir of life forms that have been accumulating for longer than any terrestrial habitat can match. This is not science fiction. This is the consensus view of deep-sea biologists worldwide. The deep ocean is not a barren wasteland where a few hardy organisms cling to survival. It is a vast, ancient, and extraordinarily diverse biome that contains more undiscovered species than any other environment on Earth, and we have explored less than 5% of it. The implications of this for our understanding of life are difficult to overstate. Consider the discovery of Loricifera, a phylum of microscopic animals first described in 1983 by Danish zoologist Reinhardt Christensen. A phylum is not a species. It is not a genus or a family. It is one of the highest levels of classification in biology, roughly equivalent in scope to the entire phylum that contains all vertebrates, every fish, amphibian, reptile, bird, and mammal on Earth. The discovery of a new phylum is an event that happens perhaps once in a century, and Loricifera were found living in marine sediments, primarily in deep-sea environments, where they had been overlooked for the entire history of marine biology. They are tiny, less than 0.5 millimeter in length, and they live between grains of sediment on the seafloor. They had been there all along. No one had looked closely enough to see them. In 2010, Loricifera made headlines again when a team of researchers led by Roberto D'Annavaro of the Polytechnic University of Marche in Italy reported finding 3 species of Loricifera living in the deep, hypersaline anoxic basins of the Mediterranean Sea. These basins, located at depths of more than 3,000 meters, are saturated with salt and completely devoid of oxygen. No multicellular organism had ever been found living in a permanently anoxic environment before. The Loricifera specimens were not simply surviving in these conditions. They appeared to be thriving, reproducing, and completing their life cycles without any access to oxygen whatsoever. Their mitochondria, the organelles that all other known animals use to produce energy from oxygen, had been replaced by structures called hydrogenosomes, organelles that produce energy without oxygen, a feature previously known only in single-celled organisms. Let that sink in. Animals living without oxygen, not in a laboratory, not in an artificial experiment, in the wild, on the floor of the Mediterranean Sea, in conditions that were assumed to be incompatible with animal life. The discovery forced a fundamental reassessment of what constitutes a habitable environment for complex organisms. If animals can live without oxygen in brine pools at the bottom of the sea, then the range of environments that might support animal life is far wider than anyone had assumed. This pattern, discovery forcing reassessment, is the recurring theme of deep-sea biology. Every decade brings a finding that overturns an established assumption. In the 1950s, it was assumed that the deep sea below the photic zone was essentially devoid of life. This was wrong. In the 1970s, it was assumed that all ecosystems depended on sunlight. The discovery of hydrothermal vents proved this wrong. In the 1990s, it was assumed that multicellular organisms required oxygen. The loricifera of the anoxic basins challenged this assumption. In the 2010s, it was assumed that the taxonomy of the deep sea was approaching completion in well-studied areas. The Clarion-Clipperton zone findings shattered this assumption, revealing that even the most intensively studied deep-sea regions contained a majority of undescribed species. Each of these discoveries was not merely additive, not simply one more species added to the list. Each was transformative. Each changed the framework within which deep-sea biology operates. And each came from a habitat that had been ignored, underestimated, or dismissed as too extreme to harbour significant life. The biochemistry of deep-sea organisms is another frontier that continues to produce surprises. Organisms living under extreme pressure have evolved proteins that function differently from those of surface organisms. The enzymes of deep-sea bacteria operate at pressures that would inactivate or destroy the enzymes of organisms living at atmospheric pressure. These piezophilic, or pressure-loving, enzymes are of intense interest to biotechnology because they represent solutions to chemical problems that surface organisms have never faced. Some deep-sea enzymes function optimally at pressures of 500 atmospheres or more. Understanding how these enzymes maintain their structure and activity under such conditions could lead to new industrial processes, new pharmaceuticals, and new materials. But this understanding requires specimens, and specimens require expeditions, and expeditions require funding that is never sufficient. The microbial diversity of the deep sea adds yet another layer of complexity. The deep sea floor and the sediments beneath it harbour an estimated total microbial biomass that may rival or exceed the total microbial biomass of the surface oceans. These microorganisms include bacteria, archaea, and viruses in numbers that are staggering. A single gram of deep-sea sediment can contain billions of microbial cells. The vast majority of these microorganisms belong to groups that have never been cultured in a laboratory. They are known only from genetic sequences extracted from environmental samples. Their metabolism, their ecology, and their role in global biogeochemical cycles are largely unknown. Some of these microorganisms carry out processes that are critical to the functioning of the entire planet. Deep-sea bacteria are involved in the cycling of carbon, nitrogen, sulfur, and iron on a global scale. They consume methane that seeps from the seafloor, preventing it from reaching the atmosphere, where it would act as a potent greenhouse gas. They break down organic matter that reaches the deep-sea floor, recycling nutrients that eventually return to the surface ocean. Without the metabolic activity of deep-sea microorganisms, the chemistry of the ocean and the atmosphere would be fundamentally different. And yet, we know almost nothing about the specific organisms doing this work. The diversity of deep-sea microbial communities is so vast and so poorly characterized that microbiologists have compared it to a dark continent of biology, a realm of life as large as any on the surface, but almost entirely unmapped. New phyla of bacteria and archaea are described from deep-sea environments every year. Some of these phyla have no close relatives among known organisms. They represent lineages of life that diverged from the rest of the tree of life in the distant past, and have been evolving independently in the deep ever since. The scale of the unknown is not shrinking. If anything, it is growing. As our tools improve, as eDNA sampling becomes more widespread, as genomic analysis becomes faster and cheaper, the number of detected but undescribed species is increasing far faster than the number of formally described ones. There is a growing backlog of organisms waiting to be named, a queue that stretches into the thousands and shows no sign of getting shorter. The bottleneck is not discovery. The bottleneck is description. We are finding new organisms faster than we can study them, name them, and understand their place in the web of life. This is the deeper truth. The deep ocean is not slowly yielding its secrets, it is revealing them at a pace that overwhelms our capacity to absorb them. Every expedition adds to the list. Every water sample, every sediment core, every camera lowered into the dark brings back evidence of organisms that have no name, no description, and no place in our taxonomic systems. The ocean is not running out of surprises. It has an inexhaustible supply, and it dispenses them with the same indifferent generosity that it has displayed for longer than any record can describe. What does it mean to share a planet with millions of organisms that have never been seen? What does it mean to live on the surface of an ocean that contains more unknown life than known? These are not academic questions. They are existential ones, questions that touch on the nature of knowledge, the limits of exploration, and the relationship between a single species and the world it inhabits. Humanity has long defined itself by its capacity to know, to name, to classify, to catalogue. The Linnaean system of taxonomy, introduced in the 18th century, was a triumph of this impulse, a universal framework for organizing all living things into a single coherent system of names and categories. For more than 250 years, taxonomists have worked to fill in that system, describing species after species, genus after genus, family after family, building a picture of life on Earth that grows more detailed with each passing decade. And yet, despite this enormous and sustained effort, the picture remains incomplete. The deep ocean is the largest remaining gap, a void in our knowledge so vast that it dwarfs all others combined. There is a philosophical weight to this that is difficult to ignore. We are a species that sends rovers to other planets to search for microbial life. We build telescopes that peer to the edges of the observable universe, looking for signals of distant intelligence. And yet, beneath the waves of our own ocean, within the borders of our own planet, lies a biome so vast and so poorly understood that the majority of its inhabitants have never been named. We look outward for the unknown while the unknown lies beneath us, in darkness, under pressure, patient and indifferent. The discovery of new deep-sea species each year is a reminder of this imbalance. It is not a sign of scientific progress, though it represents that as well. It is a sign of how much remains undone. Each new species named is a small victory, a single point of light in an expanse of darkness. But the darkness remains. The ocean does not become smaller because we name a few more of its inhabitants. It remains exactly as it was, vast and largely unexplored, keeping its contents hidden behind barriers that we have only begun to penetrate. There is also a practical urgency to this question. The deep ocean is not immune to human activity. Deep-sea trawling, which involves dragging heavy nets across the ocean floor to catch fish and crustaceans, is one of the most destructive fishing practices on Earth. Trawl nets scrape the seafloor, destroying coral gardens, sponge fields, and sediment communities that may have taken centuries or millennia to develop. In a single pass, a trawl net can obliterate habitat that will not recover within a human lifetime, and because the deep sea recovers so slowly, the damage compounds over time. Areas that have been trawled repeatedly may take thousands of years to return to their pre-trawling state, if they recover at all. Deep-sea mining poses a different but equally serious threat. The extraction of polymetallic nodules from the abyssal plain would involve industrial-scale disturbance of the ocean floor over areas of thousands of square kilometers. The plumes of sediment generated by mining operations would spread far beyond the mining site, smothering organisms over vast areas and disrupting ecosystems that depend on clear, still water. The organisms that live on and among the nodules, the very organisms that surveys have shown to be overwhelmingly new to science, would lose their habitat entirely. The tragedy inherent in this situation is stark. We are on the verge of destroying habitats that we have not yet explored, extincting species that we have not yet named, erasing biological knowledge before it has even been recorded. The deep ocean is simultaneously the largest reservoir of unknown life on Earth, and one of the most vulnerable environments to industrial exploitation. The organisms of the abyss cannot advocate for themselves. They cannot flee to safer waters. They cannot adapt to the sudden intrusion of mining equipment or trawl nets. They can only do what they have always done: exist in darkness, under pressure, in silence. This is perhaps the most profound implication of the ongoing discovery of new deep-sea species. Each new species found is not just a scientific curiosity. It is evidence of a world that exists independently of our knowledge of it. A world that does not require our attention to persist, but that may not persist without our protection. The deep ocean does not need us. But we, as the only species capable of understanding and protecting it, have a responsibility that we have barely begun to acknowledge. The philosophical dimension deepens when one considers what the deep ocean tells us about the nature of life itself. The organisms of the deep have demonstrated that life is more resilient, more adaptable, and more persistent than any surface-dwelling biologist would have predicted a century ago. Life without sunlight, life without oxygen, life under pressures that would crush steel, life in brine that would pickle any surface organism in seconds, life on the bones of dead whales, in the exhaust of volcanic vents, in the cracks between tectonic plates. Life, it turns out, does not merely tolerate extreme conditions, it colonizes them, it thrives in them, it diversifies within them, producing forms and functions that expand the definition of what it means to be alive. This realization carries implications that extend far beyond marine biology. If life on this planet can flourish in the crushing darkness of a Hadal Trench, then the range of environments in the universe that might support life is vastly larger than previously imagined. Every new deep-sea species is a data point in a larger argument about the resilience and persistence of life itself, an argument that will shape how we search for life beyond Earth for generations to come. But for now, the most pressing question is simpler and more immediate. What are we going to do about the ocean we already have? The deep sea is revealing its diversity at a rate that should inspire awe, humility, and urgency in equal measure. Awe, because the scale of life beneath the waves is beyond anything we imagined. Humility, because our knowledge of it remains so thin. And urgency, because the threats to that life are growing faster than our understanding of what we stand to lose. There is a silence in the deep that has no equivalent on land. It is not the silence of an empty room. It is the silence of a full one, a room so crowded with life that the individual voices cannot be distinguished, a room so vast that sound itself loses its way before reaching the walls. The deep ocean is that room. It has been that room for longer than any record can measure, and it will remain that room long after the noise of the surface has faded. Every year, new species emerge from that silence. They come up in nets, in sample containers, in water bottles, in the DNA extracted from a liter of cold, dark water. Each one is a messenger from a world we have not visited, carrying information about conditions we can barely imagine and adaptations we have not yet learned to understand. They arrive without fanfare. Most will never be mentioned in a newspaper or featured in a documentary. They will be described in technical journals, assigned Latin names, deposited in museum collections, and filed away in databases. But each one represents something extraordinary. Each one is proof that the planet we live on is not fully known, not even close. The deep ocean is the last great frontier of biological exploration on Earth. It is larger than every terrestrial biome combined. It is darker than any cave, colder than any tundra, more pressurized than any underground mine. And it contains more undiscovered life than all the forests, grasslands, deserts, and mountains of the surface put together. This is not an estimate. This is the sober assessment of the scientists who study it, the engineers who build the machines to explore it, and the taxonomists who spend their careers naming the creatures that come out of it. We live on the surface of an ocean planet. The water covers more than 70% of the Earth. It reaches depths that exceed the height of the tallest mountain. And in that water, in the dark, under the weight of the entire ocean above, life persists, life innovates, life diversifies. New species appear in our records every year, not because they are new to the world, but because they are new to us. They have been there all along, waiting in the dark, indifferent to our ignorance, patient in their silence. The question is not whether there are more species to find. There are thousands of them, possibly millions. The question is whether we will find them before they are lost, whether we will learn their names before their habitats are destroyed, whether we will understand their place in the web of life before that web is torn beyond repair. The clock is running, and the ocean does not wait. Somewhere beneath the waves right now, in a trench no submersible has ever entered, on a seamount no camera has ever photographed, beside a vent no sample has ever been taken from, something is alive. It has no name, it has never been seen, it exists in total darkness, under pressure that would destroy any machine we have ever built to find it. And it will go on existing, in that darkness, under that pressure, in that silence, whether we ever discover it or not. The ocean keeps its own counsel. It does not reveal its contents on demand. It releases them slowly, grudgingly, one species at a time, in its own time, on its own terms. And every year, when the journals publish their lists and the databases update their tallies, the same quiet truth asserts itself. We are not at the end of discovery. We are at the beginning. The deep has more to show us than we have years to look. And the dark water holds its silence still,